And of course, if you look at the literature, there are different areas of research going on. We all know the pesticides have a role to play. We definitely know genetics have a role to play. Bees have been bred for honey or have bred for their behavior, but not necessarily bred for their resistance, their disease resistance. And that, of course, is something different in other animal production systems where disease is the number one breeding criteria. And then, of course, you get the sort of healed side of things. So today I'm going to talk about an area of research that that James wanted me to look on in terms of the role in which the viruses play in honeybees, and he wanted me to expand a little bit on work that I used to do on superinfection exclusion, and I will do that. So it'll be the mainstay of my talk. But just to give you some background, I now based in Minnesota. I used to be based in the UK, where I've been working on mainly viruses associated with non-model organisms. So, and then I started to work on honeybees. And one of the main drivers for working on the honeybees was when I met Ron Hoskins, and this talk is dedicated to him because he was a key factor in me thinking differently about beekeeping. I was always taught as being part of the professional establishment about how one should keep bees, but his life experiences with the bees and the way he saw bees and how he believed bees could fix themselves and how he let bees develop natural resistance was something that was an eye-opener to me. And so this is sort of the work that really allowed me to look at what he was doing, the main information I gained from him, and how that had changed the way I look at viruses in honeybees. So my move to Minnesota allowed me to look at and expand the areas of interest. And just as the slide just represents the sort of breadth of research that I do now, I'm very much interested in food security. So honeybees, of course, are very important in that. Without pollination services, the sort of choices we have would be very dire. So, that's very important. But of course, animals themselves are also source of food. But in the end of the day, they all have viruses that infect them. And so my role in the vet school here in Minnesota is to try and support the veterinarians in their questions about viruses and virus diseases. I list a few viruses here. Obviously, today I'm mainly focusing on deformed wing virus. But I've worked in the past on viruses that are actually very closely related to some animal viruses, like this algal virus, which infects pigs, or viruses infect seaweed. All of these viruses have a way in which they control populations. They also have a way of interacting with each other. And this interaction between viruses, especially closely related viruses, are not unique to deformed wing virus, which I'm going to talk about more now. But all these other viruses have this role to play. And for me, having a broader understanding of virology, understanding how different systems work, allows me to answer questions and address questions in a slightly different way. And I think that my counterparts do in honeybee research. So let's go back from the beginning, right? So a lot of the attention in terms of the new attention we have on honeybees and honeybee diseases really stems from that colony collapse disorder that occurred early in the 2000s. But yet, if you speak to beekeepers, they knew for a while that the honeybee was in trouble. There was lots of issues and diseases associated with honeybees. But of course, again, became very much in the top of our mind the fact that honeybee populations are always under threat. We continually have a problem with keeping the population stable. And in fact, we do, because commercial beekeepers, and Randy being one of them, due to their breeding exercises, due to their the way in which they continue the line, especially through artificial insemination, this is what allows the bee population to remain stable. Without this, I mean, there's no other industry that can sustain 40% losses. It's without, it's the sort of breeding effect that we have within the commercial beekeepers that allows us to keep a steady population. But still, nonetheless, every year we suffer losses, 40, 50% bees. And it's not only unique to the US. In the UK, where I used to work, it's not as bad as here. It's 20% on average, 15% now, and although it looks as if it's getting better in the UK because the trend is sloping down, whereas here in the US it's pretty much steady. It hasn't improved and hasn't gone worse. Some would argue. And of course, if you look at the literature, there are different areas of research going on. We all know the pesticides have a role to play. We definitely know genetics have a role to play. Bees have been bred for honey or bred for their behavior, but not necessarily bred for their resistance, their disease resistance. And that, of course, is something different in other animal production systems where disease is the #1 breeding criteria. And then, of course, you get the sort of healed side of things. And of course, our environment. We know our environment is changing. We know we need to do better in terms of our habitat. So all these factors play. And of course, the one on the top right here, pests and pathogens, are still, for me, the #1. And it's the reason why I think it's #1 is not only because I'm a virologist, but because genetics, pesticides, and environment has been coming on for a very long time. And the sort of changes we're seeing now in the losses of our colonies is something that's happening, and it's actively happening now. It's not something that's been coming on for a very long time. So I do think the sort of disease pattern that we're seeing now is definitely to do with pests and pathogens. The one virus that seems to stand out, and again, if you look at literature, if you look at the sort of research activity, this one virus called deformed wing virus is the one is the most popular and the one that's most discussed. And I can say that I'm probably responsible for this. A lot of the work that we've been doing has contributed to this resurgence in our understanding of what's going on with deformed wing virus. And this is important because this virus is known for a long time, right? It's not in the recent 2000s that we suddenly only discovered this virus. This virus has been known for a very long time and has largely been ignored. And that's probably because of the way we think we understand this virus, and the way and the tools we use to study this virus has changed our understanding of the potential this virus has. So my talk today will be focused on this, but there will be a slight shift towards the end. So to give you some sort of background in terms of what we've come to understand about this virus, as I say, has been around for a long time. If you use modern molecular dating tools, and I won't go into, I'm trying to explain how we do this, but just believe me to say that when you try and date them, we know some of the early viruses that belong to this deformed wing virus cluster or clade is around at least 300 years old. It's not a new virus. It's been around for at least 300 years. And this whole group of viruses are predate even this. I mean, these viruses are millions of years old. But this actual cluster that we know of causing deformed wings is at least 300 years old. And this paper describes how they are related to each other. And I simplify them by calling them these 3 types, A, B, and C. In fact, there is a 4th type called D, which was one of the original strains that was isolated. It came from Egypt. And that dates it even later than the 300. It's more closely related to C. But again, what I'm trying to tell you is that this virus has been around, and this virus has changed over time. The 2 virus types, type A and type B, are the ones that have been circulating more now in the US, and I'll show you some more information about that. And also this type B virus was named incorrectly, and it used to be called Varroa destructor virus because it was originally isolated from Varroa, but it's not that virus. It's actually related to deformed wing virus, which was the one which is circulating for a very long time, called type A. And again, I try and keep the colors consistent. So generally speaking, in my slides going forward, the virus which is associated with A, which is the classic deforming virus, is red. The B is the older one called the Varroa destructor virus, but we call it type B. And C in green is the one that's been newly discovered, we discovered a few years back. So these are the three ones that we mainly focus on. The tree that I showed you now it's a related tree, right? It's how we as scientists match these viruses, and it's all to do with genetics and relatedness. And we can do this using whole genomes. In fact, that was a whole genome tree. But a lot of new trees that are generated, and the majority of trees that are generated are trees based on just small fragments of the virus genome. And these small fragments are detected or amplified using a technique called PCR, polymerase chain reaction. And we all should be familiar with this now. Well, we all live in the time of COVID, and so we all know about the sort of ways we detect. So when if you had to go to the hospital or see a doctor, other than the swab you would take and put it into what I call a pregnancy kit. Do you see the red line? Do you see the blue line, or whatever line color the company was? That's a serology test. That's immunology. That's not as accurate. But if you went into the doctor or you had to go to the hospital, they would always recommend a PCR test. And that PCR test would essentially detect the genome of the virus. It would detect what's inside this virus genetics. And that's the way in which, in fact, 99.9% of all papers that work on viruses use PCR to detect. They report on the virus genetics and ask, is it present or is it not? What's important to note is that this virus, there are pros and cons to this method. The pros are that it's very fast. You can do a PCR reaction, and usually within half an hour to 2 hours, you'll have your result. For us, when we work on bees, it's slightly more complicated. You can't swab the passages of a bee. Or you could, but it'll take time. We generally harvest a whole bee. We subsample from a colony, and we use the whole bee as the source of genetic material. And from that, we do our tests. But so generally speaking, anywhere between half an hour to 2 hours, you can get a result. Compared to those pregnancy kits or those immunology-based kits, this is cheap. This is cheap. You can really have high throughput, and it's generally sensitive. So a technique like this, you can detect between 1 and 10 copies of the virus. The cons, the negative side to PCR, is that it reports only on a small part of the genome. And in virology terms, we know that these viruses are very complicated. So for SARS-CoV-2, it's the spike protein. I don't know, you might have heard this in the media. The spike protein, how that changes will determine whether our vaccines will be effective or not. And it usually reports just on one gene. We know it's more complicated than that. In fact, persistence of even SARS-CoV-2 now is a result of other mutations across the genome. So PCR will only detect a small part of the genome and a limited information about that virus. For deforming virus, we use a different gene. This I've just put in here for those who are interested. It's the RNA-dependent RNA polymerase gene, but I won't go in more detail other than saying it's a slightly different genetic code than, for example, SARS-CoV-2. What the other downside of the PCR method is that it only tells you whether the genome is present. It doesn't necessarily tell you whether the virus particle is present. It doesn't even tell you whether the virus is infectious or not. All you know, it's there. And so I always smile when I read manuscripts about this virus, when they talk about them and they use PCR detection and they extrapolate, especially when they only use PCR to extrapolate what the abundance of this virus is. It actually doesn't really tell you a lot about whether there's still infectious material there and what the infection could look like going forward. And the other negative thing is actually, it's positive, the sensitivity side of things, is that we could be detecting contamination. And contamination, I'm meaning, is the virus could be present in a colony. The virus could be the presence on the surface of a bee, but it might not be infectious. It might not be the virus inside the bee. So your colony could be tested positive, and you will look through your colony, you'll never see disease. And your PCRs always come back as positive because it's very sensitive, but there's no virus in real terms. There's no infection going on. So again, there's positives and negatives to this test. And I'm mentioning all this because this is information that you'll need to know as I'm going through my talk today. So again, just showing you here, the deforming virus genome has about 10,000 bases, 10,000 nucleotides. It's a very, very simple genome. It encodes for genes that make a capsid, the virus capsid that we know what it looks like when you think about a virus. And then the second half of the genome just encodes for genes that allow for the virus to make more of its genome, allows for virus genome replication. See, a very, very simple virus, and yet it can do so much damage. And so when one also thinks about this virus genome, one you got to remember that these viruses don't encode for genes that causes paralysis or deformed wings. That actually it's the effect of the virus replicating that causes deformed wings. There's no gene on this virus that actually codes for deformed wings. It doesn't exist. It's the way it infects. And so it also means that you can have this virus and you can also not have deformed wings. The presence of the virus is not dictated by the disease outcome. Just like SARS-CoV-2, you can be infected with SARS-CoV-2, but you will not show disease symptoms, right? You can be asymptomatic. And in fact, in insects, especially honeybees carry viruses and they more often than not asymptomatic. They do not show disease. Again, I don't want to digress too much in this, but just say that when we do our PCRs, we only detect this RNA polymerase part of this genome, and we miss the rest. Okay. So why is this important? Why am I highlighting all this? Because there are lots of papers who are reporting this changing in deforming virus. Okay, and I'm going backwards in chronology now. So a lot of the recent papers will talk about how deforming virus A, like I said, deforming virus have different groups. The deforming virus A is decreasing over time, and deforming virus B is increasing over time. And I highlight this paper here that talks about how this could have a negative impact on bee health, because in a box here, that last point of their comments in their paper, they say that if it gets replaced, like A is being replaced by B, it could actually make it worse for colony loss and for this virus to move to other insects. Okay, because you got this changing dynamic. And let's say I think it's fair to say that this is the current storyline that you hear amongst virologists or in beekeeping communities, that there's the threat of the new deforming virus B. Eugene Raboff and Jay Evans went back to the Bee Informed Partner Survey and went to ask about this deforming virus B. And this is I took from the website, from the Bee Informed Partnership website, where they looked, and again, here you see VDV1, this is actually deforming virus B. This is the second group of deforming virus. When you look back in the history in the US, it was hardly found, right? Some areas are negative. Many states had no virus detected, and only a few states had some back in 2010. More recently, it's everywhere. And as that previous study said, this virus is increasing in abundance here in the US. Just to say, this is not always true elsewhere. Here's a virus in South Africa, a test that Steve Martin I used to work with quite a lot. He went to South Africa and did some screening in South Africa, and we knew for a long time that deforming virus B was the resident virus in Africa, especially South Africa. So back down in 2004, this pie chart here, again in blue, many places, many bees in South Africa had only deforming virus B. There was no A around. So it's not universal that A decreases and B increases. Actually, in some places, it always had B. And disease in South Africa was very rare, actually. It's hardly ever reported major colony losses. There were other interesting features that was causing problems in South Africa, nothing to do with viral diseases. And just looking at this recent study here, this paper was published in 2020, there has been a new discovery of Deformed virus A present in South Africa. And actually now there were reports of colony losses in South Africa. And this comes back to what started this whole interesting discussion about deforming virus A and B and the fact that deforming virus was responsible for a lot of colony losses was a study that I did with Steve Martin in Hawaii, where we got together and Steve went to Hawaii, collected samples, and he brought it back and said, Declan, can you tell me a little bit about the viruses there? And so we did some analysis and we found that in Hawaii, where you had no prevalence of Varroa, so what we did find, and we did confirm for the first time, that the Varroa mite was changing a lot of the viruses present in these colonies. So here in these islands in the Hawaiian archipelago, where Varroa did not exist, the virus load was very, very low. And that's another point to remember. These viruses predate Varroa entering European honeybees. The virus was always there, but the virus was a very, very low level. And in Big Island in 2010 and in Oahu in 2009, Varroa entered and Varroa increased the virus load. And so we talk about this as biological vectors, where Varroa increased the virus load. And it was that changed how the viruses interacted with the honeybee. In addition to that, what we found was that where you had no Varroa, the viral diversity, so every wedge of this pie here shows different types of viruses, the genetics of this virus. So, and this was based on the PCRs that we were doing just for this one region. You could see there was lots of diversity. But where you had Varroa present, Varroa was selecting for only a few of these genetic variants. And in fact, it was only type A genetic variant that was selecting for. So again, you have this interesting feature. There are other viruses present, but Varroa was selecting only for A. And prior to the study, there was no colony collapse disorder in Hawaii. Now you get colony collapse disorder. Well, you get colony losses. You get overwintering colony losses in Hawaii, but it never used to happen pre-Varroa entering. But again, it was deformed virus A that was present. So this brings me on to why I'm here, really. I'm giving you all this information just to set the scene. One of the things we wanted to do was Ron, as you all might be aware, in the 80s, he lost pretty much all of his colonies. And he took the surviving colonies, those that were naturally resistant, and he bred from them. And what he found was actually the bees had their own natural hygienic behavior. And he found that the bees could detect the Varroa mite in the colony, and they could selectively uncap the brood, pull out the mites, and get rid of them. He never used pesticides. He never used any control methods. He literally just bred them. And so one of the questions I had for Ron at the time was, Ron, what about the viruses? The mites can control, the bees can control the mites, but what about the viruses? And so that led to the study and this whole analysis where we talk about superinfection exclusion. And what we did was we used PCR, like I was saying, we detected a small region of the genome. And we found that in Swindon's bees, we only found deformed virus B. And, Ron's colonies, apiary is relatively small. He only had 20 colonies. He didn't have 200, 1,000 colonies. It was a very small study site that he was working on in Swindon. So these three colonies was certainly representative of his apiary. And all three colonies they all had hygienic behavior. They were all only had deformed virus B in blue. And we tested the bees and we tested the Varroa mites. So the Varroa was present. The bees were living with it, and there was only deformed virus B present. This was different to Hawaii, right? Which I just showed you. Hawaii had A present, and the Big Island, which never had Varroa, it was only B present. And as the virus was moving in Hawaii, it was starting to go from an A in some of these places here. For example, this big island where it was Varroa negative, when it became Varroa positive, it would move into an A. The A was starting to dominate. And again, this was PCR based. So to make sure we weren't mischaracterizing these viruses and saying we're only looking at the small part of the genome, we sequenced the whole genome. So this blue map here maps to the whole of the genome. And what we found was, whether it's in the colony, the bees, or in the Varroa, the bottom track, it was essentially deforming virus B that was present the whole of the genome, not just a tiny piece. But we did see presence of A, but only a small part of A, this back end part of this genome. And so it said to us that even in Ron's bees were being challenged with this deformed virus A, and the virus was not taking hold. It was deformed virus B was present and dominant, and it was not taking in. And we knew it was being challenged because the mites, if you look at this mite plot here, we got a red line that goes across the whole of the genome. We knew that the mite had the full length genome of A, but it was not getting into the hive. And that led us to this theory of superinfection exclusion. And what we understood from Ron's bees, and what we understood from what was going on in Hawaii, that the virus diversity was really high, very low levels of virus. When Varroa came onto the scene, Varroa selected for this A type. It could have selected for B because B was present at the time, but it didn't. It selected for A. It increased the virus load. We went from, say, 1,000 virus copies to now over 1 million virus copies. And that led to death. Now we get this colony loss. Ron's bees did not select for A, it selected for B. And so as a result, his virus load, even though it was high, it did not show disease. And so this is where we were saying, from what Ron was showing, because his bees were healthy. He never had disease. He kept propagating the Varroa-resistant colonies, and he was doing well, right? But yet it had deformed virus B in it. And it was somehow excluding A. And one of the mechanisms we think it was excluding is this theory, this observation we were seeing and others were observing, was called recombination. And recombination is illustrated here. And this was done by Eugene Rehoboth when he was in the UK. At the time, he found that when the viruses were present in the honeybee at the same time, the virus genomes were exchanging. So here again, you can see the virus genome drawn out here. The front end of the virus was deformed virus B, colored B, and the back end was deformed virus A, colored red. Right? These recombinants, this exchange of a genetic material, was happening in the bee. Majority of the viruses he found was still the wild type deformed virus A, and hence there was still disease in the UK. And then we went on then with Randy's help. So Randy's a co-author on here with Steve Martin. They sent us bees, and we said, what's happening in the US? Are we seeing this genetic exchange? And yes, we were. We were seeing that first of all, we were seeing that every state essentially had deforming virus A present. And so for us, it wasn't a surprise, right? Because there's lots of colony losses that's happening currently here in the US. But of course, we found there will also be in here in the US. And so you have this active exchange of A and B genotypes in these colonies. And more recently, if you look at the new literature, the new surveillance data from the Be-Informed partnership, we're seeing that the US is now becoming more blue and less red. And I'm being political here, but yeah, it's becoming more blue, less red. The UK, on the other hand, where the prevalence is still low, so the disease prevalence is low, in terms of random colony losses, the UK has now become totally dominated by B. There's hardly ever A around, but you do get the occasional A occurring, and many of these colonies are very healthy colonies. And this arrow points out time where one colony, when we were screening this in spring and in autumn or in fall, we found that the colony went from blue to red, and we now know that this colony is dying. This colony, this apiary has suffered since we observed this. So there was evidence again to show that this Deformed virus A was a problem. So there was definitely still this interaction between this virus. Now I'll come back to the fact that even this US study was very much dominated on PCR. We wanted to dive deeper and ask, what is the genome telling us? And we also went back to the Swindon bees. And the Swindon bees, we wanted to know whether this was a Swindon-only phenomenon. Why was it that the Swindon bees went dying, where we see across the world that the bee was dominating now, not the A? And through funding through the BDI, we started to look at this. And this is going to become complicated very quickly, but I want to show you something. What we did was we took Ron's colony and we moved it away from Ron. We took it out of Swindon and we moved it to the county called Devon, where I was based at the time. And we wanted to know what was going on, and we were monitored over time. What I can tell you, over the 3 years that we moved this, the colony died. Ron's colony died. So we were saying, what was going on? When we originally got Ron's colony, it had Deformed virus B. So what I'll show you now is tables of A, B, and C. And we saw it had B, and we also knew that they had some low-level presence of A and C, which was surprising because when we originally did our studies, we only found A. So what we did was we designed a second PCR study to look at the capsid genes and the other part of the genome, and said if we could find both, then we knew that the whole genome was present. And so we found, well, actually there wasn't A or C. There was actually recombination going on, again supporting this superinfection exclusion, that actually only the wild type, the whole full-length genome of B was present. So that was 2014. 2015, we started seeing, oh my word, when we moved it to Devon, we were starting seeing the full-length A take over, right? So we not only had the B present, we had lots of A for the first time, and it wasn't in Swindon, it was here, it was in Devon at the time it was happening. And what was interesting was when we went back to Ron's colony, he said, okay, Ron, we need to grab your colonies and we're going to move it to a different place. And this place, which was different from the one I described, was this place was isolated. They had no other colonies around, right? There was no possibility of an introduction of another virus. And we monitored it, and we were surprised to see that it survived for 2 years, not a problem, but we started to see full-length A present. So we went, what was going on? It made no sense. If A was present, should it not cause disease? To cut a long story short, we played around with our PCRs. We did a new assay, which I can't describe, I don't have time, just to let you know that actually this presence of this A was not really present. The PCRs were misleading. We did a new combination of PCRs and we found we weren't finding full-length A. What we were detecting was recombinant. This event here was a recombinant. So this proved our superinfection exclusion theory that actually the B virus here was recombining with the A, and there wasn't genuinely A. The A was negative. For the dead colony, which was suffered overwintering colony loss, there was full-length A. These red PCRs were confirmed to be actually full-length genome. This is an AA combination. So when people test for PCRs, they think they might be testing a full-length genome, but in fact, here we were testing recombinants. We were testing this exchange of genetic material, which then, of course, the virus behaves differently. And also, it tells you whether there's active superinfection exclusion going on. So I'm going to quickly jump ahead to say, well, actually, the story becomes even more complicated because we had that new assay. And so what we wanted to do was use a new tool that we've developed over the last 5 years that I've been in Minnesota. I've developed a sequencing tool that allows you to give you genetic sequence, whole genome sequence, as fast as PCR. So we want to develop a tool that we could take samples, whether it's animal samples or in our case, bee samples, We use robotics, high-through robotics. We create our sequencing pool of genetic material, and we do bioinformatics to sequence it. And what we found was that we confirmed our diseased bees were actually, when they were dying, they not only had B present, they had the full-length genome of A present. So the sequencing is a far better way than the complicated PCR combinations we were running to quickly determine what virus was present. Was it recombinant? Was it an active infection going on? Was it the full-length genome? And this is something that we've been doing, and this is all data that's currently being written up. So we are going to start publishing on this. And so we started to create the surveillance program, which I started here in Minnesota. We've been sampling since 2021, and we've been looking at virus genomics, looking at all different samples. And we don't only detect deformed wing virus. Here's one of my wedges for deformed wing virus. We now regularly detect acute bee paralysis virus, black queen cell virus, and this is in healthy honeybees, by the way. We aren't detecting just diseased bees, we're looking at healthy bees. And when you summarize this data, we can then start seeing that, yes, we get this new phenomenon of Lake Sinai viruses being more prevalent. In fact, Lake Sinai, these paralysis viruses are probably equally present as we see deformed wing virus. Sacbrood is interesting, is making a comeback. There's lots more Sacbrood. And this is, again, whole genomes we are talking about. And we also discovered a brand new virus in honeybees. I'm just calling it the creeper virus now. The more information will come later. But using a genomic approach, we are seeing new viruses, whereas we weren't seeing this in the past. And this deforming virus, the majority of them, we're finding them are actually recombinants. We are finding recombinants, and we are finding deformed virus B present in colonies, not A. And just as a final way to show you how the story gets even more complicated is that when you start looking at other species, we were starting to then screen not only honeybees, we were screening bumblebees, because everybody's saying that honeybees are a threat to bumblebees because honeybees are transferring the diseases to bumblebees. The early indication we're getting from the genomics, not PCR, because PCR can be misleading. The genomics are telling us that actually, no, that there are not a lot of viruses from honeybees going to bumblebees. In fact, bumblebees have their own viruses. And these viruses here that I circled came from bumblebees. These other ones at the top here are from honeybees. They are very, very different. In fact, we are finding that there are unique viruses in bumblebees that could be causing the death and the population decline in bumblebees, and they shouldn't be blaming honeybees for it. They have their own problems. So just to conclude, so you can give you some time to ask questions, is that we know that even if you control for things like pesticides or genetics, right? And well, genetics, there's the role genetics can play in controlling viruses, like the story of Ron's. The key is controlling Varroa, the vectors of this virus called deforming virus. We also know that be careful on the molecular approach you use, okay? Molecular approach will allow you to detect this virus, but also allow you to think that you have a disease that doesn't occur. Because we know, for example, bees can have deformed wings without the virus, right? And sometimes you will say, oh, there's the deformed wings and there's the virus, but actually the virus detection system is too sensitive. And so you've got to really be careful. A third point is about do not just go on PCR data. There has to be a combination of data that allows you to draw conclusions. Deformed virus B is still, deformed virus load is still important. If you have lots of this virus, you'll still have a problem. But it depends on the genome you have. And we're starting to see that the phenomenon of exclusion is actively happening in the US. We see a lot of recombinants. Some recombinants actually could still be bad, but that's another story for another time. But most of the recombinants are showing evidence that there's exchange happening between A and B. And this is why we're seeing a lot of B present in our colonies here in the US. And the other key point to make is the other important contribution that Ron Hoskins made to the field is that he actually produced a set of bees that was robust. He had a record of them. He knew what they were doing. That, you know, he was so he had I could talk provenance. He understood his bees. He kept them, and so when I worked on them, I was absolutely certain I knew what I was looking at. And this cannot be said for all bee sources, right? So I encourage all of you, if you're beekeepers, to keep records, keep track, understand your bee phenomenon, understand what is being detected, understand how it's being detected, because the information we glean is only sometimes a small part of the story. So the better the data sets, these are the data sets I'm interested in, and these are the data sets that I like working on. And again, from those genomic snapshots, these surveys that I've been showing you that have just been generated, that actually deforming virus is not the only story. And people are analyzing data now and they're saying it's deforming virus, it's deforming virus. There are so many new viruses out there that we are missing the big picture. And a lot of new viruses are coming in. And, you know, things like Lake Sinai, Sacbrood, and say, is coming back. These all have to be seen in the context of deforming virus. And I am not of the opinion that deforming virus B, the resurgence of B, is causing the damage we are seeing now here in the US. So I'll leave it there now. If there's any questions, I'm happy to take.
Thanks, Declan. Thank you very much for the presentation. I've got a couple of questions that are queued up. And then once we get through the queued up questions, if we have an appropriate amount of time left, without stepping on too much of everyone's schedule, we will allow those individuals to just go ahead and unmute and take the question directly, if that's okay with you.
Absolutely.
Okay, so the first question that popped up was earlier in the talk. You said you can have DWV but not see visible DWV in the bees themselves. Does the prevalence of the DWV have an impact on the external evidence of DWV? So if there's more of the virus, are you going to see more occurrence of the shriveled wings?
Absolutely not. You don't have to. That's a very important question. They're not correlated. Just because you have, you know, 100 million viruses of deforming virus present in your bees does not mean you'll ever see symptoms. Doesn't mean you'll actually see the symptoms. It's actually never correlated. In fact, the best correlation is with the presence of Varroa. If you have Varroa plus the virus, you'll get symptoms. You'll see more often the symptoms. But you can control your Varroa. And I've had many colonies where we see no Varroa present, and you have more than 1 million deforming virus particles present. So its amplification can be done through other routes other than the Varroa itself. And so, yes, so then the question arises, what's the role of this virus? It's not only deformed wings, it's other things. It's like colony loss. It's changing behavior. These are the factors that we need to think about and not the deformities.
So if I were to tease my friends who enjoy treating their bees and I say, hey, see, you treated anyways, but you still have deformed wings, so obviously you have a problem, it may actually just not be true. That's not necessarily rooted in fact. It's not a direct correlation. If you've wiped out the mites and you did treat effectively and you've controlled the population, you could still have an occurrence of the deformed wing, and I have no grounds to tease them.
Yes, don't tease them.
All right.
That virus can come. And the thing is that virus can be present anyway. And the other thing is also who's been telling them the virus is present. So if you use PCR, the PCR can also be misleading because the virus can be present but not infectious, right?
So there'd be a greater degree of worry if I'm seeing both along with the mites.
Yeah. So you have to have the big picture. You need to have a good history of your colony to understand when these occurrences occur. But again, the important point is don't rely on that symptoms, deformed wings. It's a one aspect of what this virus does. It's not the big picture. So the combination of what's going on holistically in your colony, that together will make a decision whether deforming virus is playing a role or not.
We're taking all the facts, not just the feelings.
Right.
Yeah. Okay, so this one was related to the Swindon bees. And did the bees that were moved swarm between 2014 and 15, or was it the original queen when you brought them to Devon?
It was the original queen. We literally put the whole thing in a so we had a beekeeper did that for me. Took the whole colony, wrapped it up, and brought her across. So, and you're right. More often now, when you do this, you get swarming events. Now we monitor that queen. The one that was healthy, actually, it did change queen over time. So that colony that went down, it was in the first year and a half that it went, because the deforming virus A was very aggressive. The other one, over time, she did swarm, and in fact, we went Ron, right? We went like Ron would go and change and make sure he put a new queen with hygienic behavior. We let it run, and we kept the whole colony straight from Ron's bees, and we moved it.
And Ron was a big proponent of resistance breeding. I think it was just maybe a year or two ago that he had passed. Is that correct? I think it was
2022.
Not too long ago. So this question is, what would you speculate as a major contributing factor of honeybees succumbing to the host of diseases you've discussed today? So like, what are some factors beekeepers can evaluate to move ahead while still reducing their chemical dependence? And are there bees in the genome that we could seek out to improve our chances?
Right. So what I would suggest is good practice. So in other words, what I'm saying is, if you have multiple colonies on one location, right, try not to cross-contaminate. Don't work with one colony and then don't go and, you need to wash your hands. If you're wearing gloves, change your gloves. Then go into the next colony. It's the spreading. You actually have a really good role of moving viruses, just like we do with human viruses. You can move bee viruses from one colony to another. So try and keep locations separate, colonies separate. That's one of the best things you can do. Do not cross-contaminate.
Would DWV cross-contaminate, like along with hive materials, frames, maybe swapping things over, other types of equipment transfer? Would those also maybe create inter-apiary or even inner-apiary spread?
Yep, absolutely. Because this one of the key things about this virus is very, very stable outside the bee. It's stable outside Varroa. It will be on the surfaces. Now, that's another thing. Just because you move it across doesn't mean that's going to automatically go and infect the bee itself, right? Because there's a natural occurrence of this virus now. It's pretty much everywhere. So there's still an active transmission route, and we still don't know how that would work. Is it pure prophylaxis? Is it just purely the bees cleaning themselves? What is the main avenue on which this virus would then get into the colony once you've moved it into the frames, into the wax, into all the materials that you use? Yeah. So try and keep your tools separate, or at least wash them. So alcohol washes, wash it with alcohol. Try and use disposable tools if you can. But yeah, we are probably the biggest culprits in moving viruses around.
Yeah, me personally, I really try to wash all of the hive tools. I have one for each apiary, or two if I lose one. But even on a day where you're really busy, you might grab one tool and you didn't have time to clean it, and you got to get stuff done, and it could actually have a very detrimental effect. So being hygienic with your tools is really a big help.
Absolutely.
Declan, one of our board members, Matthew Kobe, has a question. He's asking to unmute. Did you get unmuted there?
Yeah,
Matt?
I think so. Can you hear me?
Okay. Yep, we can hear you, Matt.
Hey, Declan. So you mentioned at the very beginning of your chat or your talk about how these viruses have existed pre-Varroa. And so I guess my main question is, for the recombination to occur, do you suspect that the recombination has been occurring throughout the years and we are just now noticing it? Or do you think there's something to do with Varroa that's allowing the recombination of A and B?
Yeah, so for recombination to take place, you have to have high levels of the virus, because the virus has to co-infect a single bee. And not only that, it has to co-infect the cell that the virus is replicating in. So the only way it happens, you have to have these two viruses together. Now, normally, if your virus abundance or load is low, the chances of that happening is rare. It doesn't. In fact, it never happens, because there's never an opportune time for both viruses to be present at the same time for the same infection to take place. So yes, Varroa has changes. It's a Varroa that's increased the virus load dramatically, right? We went from background levels to now 1 million viruses per honeybee. Because of that, the frequency of recombination has increased. But that's also an Achilles heel for the virus, right? Because the virus actually doesn't want to exchange its material like the way it's doing. It needs to, so this genetic evolution side of things, that's a bad thing for the virus to change too much. So evidence of things like recombination can have both good and bad consequences for the virus. And the majority of the time, recombination, the large genetic exchanges, has often a detrimental effect on the virus. So in fact, most of the detection we do on virus particles, they do not show recombination events. They actually show still wild type. So there's a fitness disadvantage to be a recombinant. Does it answer your question, Matt?
Yes, very thoroughly. Thank you very much.
You got any follow-ups, Matt?
I had a follow-up, but he answered it already.
Great. Okay, so I think we can do one or two more here. And this one is relative to the Sustainable Beekeepers Guild of Michigan, and our mission and philosophy is to try to reduce the dependence on out-of-state bees and raise local stock. And, when it comes to the importation of other genetic material, we want to try to reduce some of the factors that you've mentioned here. Adrian Quiney asks, given the risk, does it make sense to buy bees from out of state?
No, it doesn't. Okay.
And this will get you in trouble.
Right. So let me be very carefully clear. Yeah. Okay. So as a philosophy, I would say no, right? Because you are moving material around, and so you're moving its diseases around. Okay. So as a principle, if you can not bring in, then don't. That said, we're not living in a vacuum. We are living in a time where it's almost the genie's out of the bottle, right? The virus is pretty much everywhere. So across the US, go anywhere across the US, get your material across the US. It doesn't matter, it's all ubiquitous. We move, 80% of our colonies move across the US anyway. So for the pot of the US, United States, America, move, it doesn't matter, you're not gonna change. But again, right, if you don't have to, don't. Because you are reducing the risk of you bringing in something new that could be developed because the pressures across the US are different. So what's happening in California is different to what's happening here in Michigan. But it doesn't mean I'm saying to you, do not go buy queens from or material from California. Yeah, but it's always a risk. It has to be, right? You are bringing something new. But again, the big picture is we are in a state where everything's everywhere at the moment in the US.
Ultimately, do your best to reduce your risk as best as possible, minimize your inputs if possible.
Right.
Very good. Do you see a correlation with pesticide exposure in DWV, or is this something you can answer?
Yes. No, I've not done pesticide checks. But my a lot of the samples I take, a lot of the work I do is bees that are in the environment. So it's like your bees, right? So they will be exposed to pesticide. So all my bees will have the same equivalent pesticide exposure. So whether the controls or not, they'll have pesticide exposure. There are other studies that have looked at pesticide, and some produce inconclusive data. There are others that say, yeah, there's a potential increase because pesticides change the immune system. The immune system is affected, and then you get disease. For me, it's still a big black box. We don't know, but I don't think it makes that big a thing because all my bees are exposed to the same levels. So, we see differences just on the viruses.
Are you seeing as much competition with the DWV as this virus is mentioned in the question, AMSV1? Is this something you are aware of?
No, just rephrase that again.
What of the new virus or newish virus AMSV1 in competition with DWV?
Right. Okay. There's another proof point. So bees are amazing things, right? Because bees have so many viruses, so they will compete with each other by the infection. The fact they coexist is a really interesting feature anyway, to have these multiple infections all the same time within the colony. When you have these new viruses and these new interactions, we have no idea what they're doing. I mean, this is such an early these are still early days for us in virology for honeybees. When you consider that for a long time, we ignored viruses in honeybees. It's only this last 10 years, 15 years that honeybees and honeybee virology has been taken seriously. So their interactions, no, we're not even close to it. But I'm hoping to do that, right? With my sequencing tool, I'm measuring everything now. And so I'm not just focusing blinkers on a deformed wing, I'm actually incorporating all the others.
Matthew, did you have another question? You can unmute yourself, I think.
Yeah, but it's kind of going back to the correlation between you said there is no correlation between the viral load and basically the symptoms of DWV. Did you ever find bees that carried a low viral load but were still symptomatic?
Yeah, there are examples. There are other reasons for why a bee has deformed wings, right? So because remember the regional characterization of deformed wings is where they actually injected bees and then they left it and they found that the bees with high load just don't, when they mature, the bees don't expand their wings, right? They don't push the hemolymph, and so you get the expansion you get. So that's why, so this is why a fully developed bee with fully developed wings can have deformed wing virus, because it's ready, the process of making the wings are already over, right? So the reverse is also true. You can have deformed bees, but they deformed for genetic reasons or for other reasons, for the inability for the wings to expand. It doesn't have to be because of virus load. And then you can have the associated virus, which is there at low levels, and you won't get wing deformities. And in fact, one thing that Steve Martin always told me, he said, Declan, remember, if you have a very high deforming virus load in pupae, the source of the deforming bees, those pupae will die before they produce deformed wings, right? So the actual real role of the deforming virus at high levels is death of the pupae, not deformed wings only. So that's why there's no correlation.
So that susceptibility could be evolutionary gain then by the bees, because what you're saying is that generation will die out and take the virus load with them.
Oh, let me tell you, viral ecology is only in the infancy with this. There's so many natural processes at play that the problem is, of course, bees are so important. Bees are pollinators for us. So we have to find ways, humans, to interfere. We want to find a way to solve the problem because we think we can solve all problems. But over time, if you let bees themselves, you're right, that's a genetic disadvantage for the bee. If the bee has very high levels, it'll kill the actual developing pupae, and the worker will remove it. And so you would actually have a way of removing it. But of course, we are trying to interfere. We're trying to do this, and we'll, in the early periods, we'll cause more harm than good. But eventually, we'll hopefully find a solution. But if we leave the natural evolution to take place, there'll be a lot of death. There'll be a lot of destruction, and that's part of the problem why we in now, because we haven't, the solutions we think we are applying is not good enough. So yeah, there's natural evolution going on, and it's really complicated, and we are trying to decipher this in a super colony. Who knows?
Well, Declan, I feel like this went by way too fast, and I wish we had a lot more time. It's been a great discussion, and I appreciate your presentation and everything that you've shared with us and the questions you've taken. It's a lot to take in.